Good afternoon, everyone. Thank you for joining us. Welcome back. I'm Steve.
And I'm Scott.
And together we are Backyard Musings, the Science and Technology channel.
Yep, and we are talking, finishing out the day with another health topic. So we hope you enjoy it.
A little known gene turns out to be vital for insulin production in the earliest stages of life. Recent advances in DNA sequencing combined with new stem cell research approaches have allowed an international group of scientists to identify a previously unrecognized form of diabetes that appears in infancy.
Oh,
The research was presented in the Journal of Clinical Investigation on September 9th last year. Would you like to know more?
researchers at the University of Exeter Medical School, working alongside colleagues at the Universitat Libre de Bruxelles. I don't know if I'm pronouncing that at all right. We're short for ULB in Belgium, and other institutions determined that changes in the TMEM167A gene, which, I mean, what do we got to have? 4 or 5 of those in our body probably, are responsible for this rare type of neonatal diabetes. Diabetes can emerge in some babies within the first 6 months of life. Wow, that's
Crazy.
crazy. And in more than 85% of these cases, the cause lies in inherited genetic mutations. In a study led by the University of Exeter, scientists examined 6 children who also had neurological conditions such as epilepsy and microcephaly,
Uh-huh.
and found that all shared alterations in a single gene, TMEM167A. So,
And look, I just want to add, folks, that tomorrow we're going to discuss an article about how 8 babies were created from DNA from 3 parents. You heard that right, 3. Um, and it's to weed out all this stuff that could happen. So join us tomorrow morning. We're going to get to that.
oh,
To
are we
click,
trying? Are we trying to make a perfect race? Is that what you're saying?
trying to what?
Make a perfect race?
And that's what it sounds like. One
Yeah,
without diseases, I guess. Yeah.
yeah. There we
Uh,
go.
to clarify how this gene functions, Professor Miriam Knopp's team at ULB used stem cells that were guided to become pancreatic beta cells and applied gene editing techniques, CRISPR. We've talked about CRISPR,
Oh yeah,
CRISPR.
yeah.
Their experiments showed that when TMEM's 167A is disrupted, insulin producing cells lose their ability to function properly. As a result, these cells trigger stress responses that ultimately lead to their death.
Dr. Alyssa DeFranco at the University of Exeter said, finding the DNA changes that cause diabetes in babies gives us a unique way to find the genes that play key roles in making and secreting insulin. In this collaborative study, the finding of specific DNA changes causing this rare type of diabetes in 6 children led us to clarifying the function of a little known gene, TMEM167A, showing how it plays a key role in insulin secretion, unquote.
Professor Knopp said, quote, the ability to generate insulin producing cells from stem cells has enabled us to study what is dysfunctional in the beta cells of patients with rare forms as well as other types of diabetes. This is an extraordinary model for studying disease mechanisms and testing treatments.
This discovery shows that TMEM167A gene is essential for the proper functioning of insulin producing beta cells, but also for neurons, whereas it seems dispensable for other cell types. These results contribute to a better understanding of the crucial steps involved in insulin production and could shed light on research into other forms of diabetes, a disease which today affects almost, this is staggering, 589 million people worldwide. That is
And,
just,
and remember, this morning we talked about how this diabetes could lead to heart disease. This is staggering worldwide, staggering.
that's almost twice the population of the United States.
Yeah.
That is crazy.
All right,
Crazy.
folks, thanks for joining us. We'll catch you in the morning.
Take care, everyone. Have a good day, rest of your day. Thank you.